Hello and welcome to Chapter 12 Part 1B on Aerobic Respiration. Now, as we spoke about last video, aerobic respiration is the breakdown of organic molecules to release energy, which is used to synthesize ATP. And because of the word aerobic here, it is in the presence of oxygen. Now, I just want to highlight to you that, hey, there are many different kinds of organic molecules here that can be broken down to synthesize ATP, but we primarily focus on what glucose is doing because, frankly speaking, that is the easiest one and the least complex out of the four. Now, there are 4 stages in aerobic respiration of glucose that we're going to finish in one video today. So this is going to be quite a lengthy video, I think. I'm going to try to be as precise and succinct as possible. So these 4 stages are here. You do need to know the names by heart. So before we start now, I think what would be helpful if you knew the 4 names. If you have not memorized it, please pause this video and think to yourself. Repeat these names to yourself 7 times over. It really helps. Also remember what location that occurs in. Glycolysis occurs in the cytoplasm, link and Krebs in the mitochondrial matrix, and oxidative phosphorylation in the inner mitochondrial membrane. This is 4 stages, but each of the 4 stages actually has more stages. In real life, oxidation of glucose in cellular respiration is not just this very simplified version of a chemical equation. It is a multi-step reaction. Even glycolysis, which is the first stage of glucose oxidation, looks like this. There are many steps in between. Each step would release a small quantity of energy. And I guess the question is, why do we want to do it step by step? Can't we just have one step and get it all over with? Well, the more steps we have, it allows us to have precise control over what is going on. We can stop it, we can accelerate it according to what the body needs. Secondly, even if all the energy, so 2870 kilojoules, all this energy is made available in one go, the cells could not fully harness the total energy release. So there's no point in trying to finish it as quick as possible. And also, there might be a lot of wastage when total energy, the energy release is not used up. And also, there's one more reason, is that reactions do not happen that easily. It requires many steps for it to happen, and the main reason for that is because glucose is quite a stable substance. Look at it, it's a ring form. There are like 5 carbons in a ring, 1 sticking out. It's quite a stable substance, okay? And it requires a very high activation energy for the reaction to take place and for it to be oxidized into carbon dioxide and water and energy. So when you hear activation energy, I hope you are thinking enzymes, because what we're going to do is to use enzymes to lower the activation energy in order we can break down glucose. Now, not only one enzyme is needed in this, many enzymes are needed in the different processes as well. The second thing we can do is to raise the energy level of glucose by making it more, by making the energy levels higher. We can actually speed up the rate of reaction. Now, what do we mean by phosphorylation? We mean adding a phosphate group to the glucose molecule in order to carry out the reaction. We will see an example of this in the first stage, which is glycolysis. This is stage 1 glycolysis, aka the glycolytic pathway if you're feeling fancy. Here are the steps. Number 1, glucose, a 6-carbon sugar, is phosphorylated. So this is glucose here, this is a representative diagram. What happens is the ATP comes and it is phosphorylated. And it is phosphorylated again. So here you need 1 and 2 ATP. What you result with is fructose bisphosphate, or another name for it is hexose bisphosphate. It is a 6-carbon molecule with 2 phosphate attached at the ends. And what this does is that it raises the chemical potential energy of glucose, and this provides the activation energy for the split. Split into what? Split into 2 triose phosphate. 2 triose phosphate are the same, yeah. they both have 3 carbons and 1 phosphate group. These 2 molecules are the same. It's just split into half. Now what happens next is that 2 hydrogen atoms are removed. In the process, 2 ATP is also formed. Now what happens to 2 hydrogen is that NAD, this molecule that you have never heard of, comes along, okay, and removes the H and becomes reduced. So NAD is reduced, so we call it 2 reduced NAD form. So NADH and reduced NAD are the same thing, okay? This is an oxidation reaction for the triose phosphate. Yeah, because 2 hydrogen atoms are removed from triose phosphate. So triose phosphate is oxidized, or we can say it is a dehydrogenation reaction because 2 hydrogens are removed. After that, it's converted to some intermediates that we don't need to know about, thank God. And then in the process, another 2 ATP is formed. So as a result, you can see here, 4 ATP is formed. But since 2 ATP has been used in the beginning, so we said that glycolysis has a net gain of 2 ATP. 4 ATP is produced, 2 ATP is used. So a net gain of 2 ATP. And at last, the final product of glycolysis would be pyruvate. When the 2 phosphates have been removed, it is just a 3-carbon molecule. Just in case you haven't noticed, the number of carbons is very, very important. So for every molecule, remember the name and remember the number of carbons and what happens to it before and after. So glycolysis, what's going on so far? There is phosphorylation. Okay, phosphorylate here and phosphorylated here, forming fructose bisphosphate, and then it breaks apart. So from this area to this area, there is a lysis reaction, and after that, 2 hydrogen is removed and ATP is formed, and this area here is called oxidation or dehydrogenation. And we say that this ATP here is synthesized to something we call substrate level phosphorylation. So this is individual molecules phosphorylating each other and forming ATP. Okay, so this is substrate level phosphorylation. So let's look at a summary of this again. So quick recap: glycolysis occurs in the cytoplasm. There is the lysis of glucose from a 6-carbon into a 3-carbon pyruvate. It involves many enzymes at each step, but you don't need to know the enzymes here. It is multi-step, which means there are actually more steps than these. The initial reactants are very important. Initial reactants are glucose and 2 ATP. And if you want to be meticulous about it, 4 ADP used to make ATP and 2 NAD here. And what the products are per molecule of glucose is 2 pyruvate, which are 3-carbon molecules. And of course, don't forget the 2 NADH formed here, as well as the net gain of 2 ATP. Again, 4 ATP is produced, but 2 are used, so net gain of 2 ATP only. So that is glycolysis. Okay, let's pause and talk a little bit about hydrogen carrier molecules. You've seen the hydrogen carrier molecules in this area just now. What in the world is NAD and where did it come from? What is its structure? What does it do? We are about to find out. Now, hydrogen carrier molecules or hydrogen carriers are also called hydrogen acceptors. You'll learn 3 types in A2. You'll learn NAD, which stands for, brace yourselves, nicotinamide adenine dinucleotide, which is used in respiration, NADP, which is very similar to NAD except there's a phosphate behind, and this is used in photosynthesis, which we will cover next chapter. And number 3 is FAD, which is also used in respiration, and it stands for flavin adenine dinucleotide. We will see FAD very soon in the same video. Now, they are all coenzymes, which means they are non-protein, they are not made out of protein, and they are required for the enzyme's activity. So let's talk about NAD first. Again, it is nicotinamide adenine dinucleotide, and this sort of gives you a clue into what exactly the structure is. Again, it is a coenzyme. Enzymes need it in order to work and function in glycolysis. And it's a hydrogen carrier molecule in respiration. Now the structure is that it has two linked nucleotides, dinucleotides, but not a phosphodiester bond, or there's no sugar phosphate backbone or anything like that. It is bonded like this at the phosphate group, to the phosphate group. Okay, both have ribose sugar and a phosphate group each, but one has a nicotinamide ring, which is a group here, and the other has an adenine base, and therefore it's called nicotinamide adenine dinucleotide. Get it? So the nicotinamide ring actually can accept hydrogen. So in glycolysis, when it receives that hydrogen, it attaches it here. Okay, you don't need to know the particular chemical reaction, but you do need to write this ones down here. NAD plus H will equals to reduce NAD, or you can express it this way: NAD plus this positive here plus H plus 2 electrons would equal to NADH. Now, what is its function? Now, it is a hydrogen carrier molecule in respiration, and actually a lot of NAD is formed from all stages of respiration. Stage 1 is glycolysis, stage 2 link reaction, and stage 3 is Krebs cycle. And after that, it will take part in stage 4, which is oxidative phosphorylation, where most ATP is synthesized. So that is NAD, and we will learn more about it as we go along. Don't worry about it. Let's talk about NADP. Now, basically, it's the same thing, but plus an additional phosphate. So you can see here NAD, 2 ribose nucleotides, one with a nicotinamide ring, one's an adenine. This is pretty much the same, except that there is an extra phosphate group at the end. It is a hydrogen carrier molecule as well, but it is used in photosynthesis. It is not used in respiration, which means you don't have it, plants have it. Now, let's talk about FAD. FAD is different. It is called flavin adenine dinucleotide. The only sort of same thing is the nucleotide part, which it's not very obvious here, but both of these are nucleotide variants. They are types of nucleotides. You can see here that it is again a coenzyme. It is a hydrogen carrier molecule also in respiration, just like NAD, but it's completely different. Yeah, different structure completely. And what it is used to do is to carry hydrogen produced in Krebs cycle and then use it in oxidative phosphorylation in stage 4. Okay, so NAD is produced in stage 1, 2, 3 and then used in stage 4. FAD is also produced in stage 3 along with NAD and used in stage 4. So there is something special about stage 4 you will learn later on. The structure is described here, even though I briefly talked about it just now. It is again 2 linked nucleotides. It says it's a dinucleotide. One nucleotide is with a phosphate, ribose, and adenine. It looks very normal. But the other nucleotide, it's strange, it's really weird. But what it has is a phosphate group, a ribitol, and a flavin, which are these 2 compounds right here. Now, we don't need to know how to draw this, don't worry about it. Sometimes you might need to identify it. It looks very distinctive, so I'm pretty sure you can do that. But most importantly, we do need to know how to write these equations. So, FAD doesn't pick up only 1 hydrogen, but it picks up 2 hydrogen, so it's plus 2H equals to reduce FAD. Note the reversible sign here. Or you can write it this way: FAD plus H plus 2H plus 2 electrons will be FADH2. Going back a bit to NADPH, which I forgot to talk about just now, NADPH is pretty much the same as NAD except that it has a phosphate group attached to it. The same goes to its chemical formula. Stage 2, the link reaction. These are the steps for the link reaction. It occurs at the mitochondrial matrix. Okay, that's important to know. So pyruvate, which is formed through glycolysis, has to be transported from the cytoplasm into the mitochondrial matrix. This is an active process. It requires active transport. Once it's in the matrix and oxygen is available, pyruvate is then converted into carbon dioxide. How? Okay, so it's decarboxylated, so carbon dioxide gets out. It's dehydrogenated, so a hydrogen atom gets removed and NADH is produced. And the third thing is, it is combined with this coenzyme A. Now, coenzyme A is not a hydrogen carrier. Coenzyme literally means that the enzyme needs it to be able to function. So, since one carbon is removed through carbon dioxide here, we are left with 2 carbons and 1 coenzyme A. Together, that makes acetyl coenzyme A. Again, the number of carbons is very important. Please look out for that. So, acetyl coenzyme A, again, 2 carbon molecules plus the coenzyme A together. Perfect. Nice. So, these are the steps when you're talking about the type of reaction that's occurring. So, this is decarboxylation again. This is dehydrogenation, or you can call it oxidation. I prefer dehydrogenation because it's just easier to remember. Removal of hydrogen, yeah. And also, it's combining with coenzyme A. There's no specific general name for that. Now again, what is coenzyme A? Here we go. It is actually a complex molecule. We usually use CoA as a simplified chemical structure, but you can see here that actually it is pretty crazy. It is made out of a nucleoside. Where's the nucleoside? Nucleoside here, which is adenine and ribose, and also a vitamin, pantothenic acid right here. So here is coenzyme A. The acetyl 2 carbons will bind right here to form something that looks like this. You can see the sulfur. This is sulfur here. What's its function? Its function is simple. It just carries the 2 carbons that we just formed to the Krebs cycle, which is stage 3. So the summary is here. Link reaction happens in the mitochondrial matrix. The initial reactants is 1 pyruvate, 1 NAD, and 1 coenzyme. And the final products of the reaction per molecule of pyruvate is just 1 acetyl coenzyme A, and of course 1 NADH and 1 CO2, which is a waste gas, a waste product of respiration, and is released. But you know, don't forget that pyruvate, two molecules of it are produced during glycolysis. Okay, so 1 glucose oxidized to 2 pyruvate. So per molecule of glucose, there would be 2 acetyl coenzyme A, 2 NADH, and 2 CO2 produced. So that's the link reaction. This is the Krebs cycle. So the Krebs cycle is also known as citric acid cycle or the tricarboxylic acid cycle, and these are names you will see in past year sometimes or in reference books, so don't be frightened. It's the same. It's Krebs cycle, same thing. And as the name suggests, it is a cycle, and this here drawn for you is actually a very simplified version of the cycle, and yet it still looks so complicated, right? I know, I was scared too when I first saw it, but don't worry, we'll work through it step by step. Okay, so Krebs cycle, where does it occur? It occurs in the mitochondrial matrix, so the same place as the link reaction. It is an enzyme controlled pathway, so please bear in mind that even though the enzymes are not shown here, they are working, like each step requires an enzyme. So let's start. The first step is this. Acetyl coenzyme A, a 2-carbon molecule that we have produced in link reaction, now enters the Krebs cycle. Ta-da! And what happens is it combines with oxaloacetate, which is a 4-carbon molecule. So 4 plus 2 will become a 6-carbon molecule called citrate. So that is oxaloacetate forming with acetyl coenzyme A to form citrate. Now you realize here that the coenzyme A is not drawn anymore, and the reason is because it is removed. Coenzyme A is removed and can be used again in the link reaction all over again. So we just kick it away. Ding. Now, what happens to citrate? Now, citrate goes through a series of dehydrogenation and decarboxylation reactions, which means, well, the hydrogen is removed, carbon is removed. And you can see here ATP is formed, more hydrogens are removed. So 7 steps here from citrate to this 4-carbon molecule, to this oxaloacetate here, and the products of each step is this. So this is how I remember it. Number 1 is nothing. Number 2, you remove hydrogen and you remove CO2. So once you remove, if you decarboxylate something, it's going to be reduced by 1 carbon. So you end up with a 5-carbon molecule. And then you do the same thing again. So you reduce NAD again and CO2 again. And you form a 4-carbon molecule. Now after this, what happens is ATP is produced. Ta-da! 1 ATP only, don't so excited. Then after that, FAD is produced. This is the first time we have seen FAD. Reduced FAD means it carries 2 hydrogens. Yeah. Then number 6 is really nothing really you can see. And number 7 here is reduced NAD that is formed. Now 7 is the number of completion, which means we are complete with our 7 steps. And it has regenerated back into a 4-carbon oxaloacetate. Now we realize here that actually by these states right here that I'm pointing at, the previous acetyl CoA in like white C's, they are already decarboxylated. So from step 4 up to step 7 here is pretty much the cycle trying to convert it back to oxaloacetate and get as many hydrogens as possible. Now what can the oxaloacetate do? It's simple. It can just, it's a cycle, right? So it combines with another acetyl coenzyme A and the Krebs cycle continues on, and this 7th step continues on and on again. Now you might be asking me, teacher, how to remember? Oh, got so many things. Now the honest answer is I don't know. I don't know how to memorize this either, but it sort of went in. The only weird way I could memorize it is by a story, a ridiculous story. And if you don't want to hear it, you can skip ahead, but if you want to hear it, pay attention, okay? So here's the dumb story. Once upon a time, there was this guy called Ox, and his full name is oxaloacetate. Okay, fine, it's not a guy, it's an animal. And this ox accidentally ate too much acetyl CoA and oranges where citrate is found, and he became a really, really fat, angry ox. In fact, he was so fat that he couldn't move the first round. But after 2 hours, he started vomiting, strangely enough, coconuts. Get it? Co-nut. So CO2 is produced and NAD is used to produce reduced NAD. And then he vomited another coconut. So CO2, NAD. Right after this, he was so tired because he has released all his energy. So he sort of took a nap. But he realized that wasn't good enough because he was still fat, right? So he needed to reduce fat. So he started working out. He worked out and worked out until he was so tired. And he thought to himself, I'm never going to do this again. But obviously, oxaloacetate, the guy, does it again and again and again all the time. So that's my dumb story. I hope it was helpful. If it wasn't, come up with your own story and remember it, okay? Moving on to actually what comes out for the exam, let's talk about the type of reactions that occur at each step of the process. So at step 1 here, there is dehydrogenation because hydrogen atoms are removed from this 6-carbon molecule right here to produce reduced NAD. Decarboxylation happens and CO2 is produced. And again, the same thing here, dehydrogenation, decarboxylation. ATP is from the 4th stage here. ATP is formed by substrate-level phosphorylation. The same word that's used in glycolysis just now for ATP production, same word here, substrate-level phosphorylation. The next stage, we have reduced FAD form. So dehydrogenation occurs as well because FAD carries 2 atoms, hydrogen atoms, away from that 4-carbon molecule. And then number 6, nothing we can see. Number 7, there is a reduced NAD that is formed, which means that there is a dehydrogenation reaction as well. So, just so you know, as we mentioned just now, there is a very simplified version of the Krebs cycle. I doubt my Ox story would benefit you in university as much because there will be so many more names to remember. It only works for the A levels, okay? Each and every substance has different names, and this is the mnemonic that they use to remember quite often, but you don't need to memorize this. And there is enzyme names at each stage. But all you need to take away from the Krebs cycle is this. Number 1, you need to remember 3 compounds. Names of just 3 compounds, not all these. 3 compounds: oxaloacetate, acetyl coenzyme A, and citrate. Okay, you need to know the byproducts formed at each step. So the 7 different steps we just talked about just now. Make any story you want, don't really care, as long as you remember them. Number 3, you need to know the number of carbons in each intermediate compound. So oxaloacetate has 4, acetyl coenzyme A has 2 carbons, and then citrate has 6 carbons. But after that, the carbon number changes as well. The easiest way to remember it is that every time CO2 is removed, the carbon number reduces by 1. Okay? Remember that each step requires an enzyme, and the enzyme is usually not shown in diagrams, and that is okay. Just remember it's catalyzed by enzyme and it's tightly controlled. So let's have a little summary here. Krebs cycle occurs in the mitochondrial matrix. It is an enzyme-controlled pathway. Initial reactants is oxaloacetate, acetyl coenzyme A, and all these other molecules. That is not important. The bold ones are more important. The final products per molecule of 1 acetyl CoA. This is 1 turn of the Krebs cycle, would be 3 NADH because it's produced at step 2, step 3, and step 7. 1 FADH2 or reduced FAD, you can use either term. 2 CO2s and 1 ATP molecule. And of course, the oxaloacetate molecule is also regenerated and then reused in the cycle. But again, 1 molecule of glucose that we had in the first place is oxidized into 2 pyruvate, which is converted to 2 acetyl-CoA in the link reaction. So for final products per molecule of glucose, it would take 2 turns of the Krebs cycle. There's a typo here. 2 turns of the Krebs cycle. So you have double of everything that is formed there. So far we have done glycolysis, link reaction, and the Krebs cycle. So if you have this in notes or printed, great, you can fill this up by yourselves. If not, you can draw a table in your notebook, okay? So these are products so far per molecule of glucose. It's not per pyruvate. Yeah, it's per molecule of glucose. So your products has to look something like this. So glycolysis, net gain is 2 ATP, NADH is 2 that is formed there, and link reaction in Krebs cycle, you can see that the information here has doubled because, well, one molecule of glucose produces 2 pyruvate molecules, and those pyruvate molecules individually go through the link reaction and Krebs cycle. So there will be 2 turns of the Krebs cycle and 2 link reactions. So in total, we have 4 ATP. Yay, we have 4 ATP. We have some energy. We have 6 CO2, which is a waste gas that is released, 10 NADH and FADH to use in oxidative phosphorylation, which is stage 4. Now we are looking at stage 4 oxidative phosphorylation. Now before we go into the steps, we need to do a little bit of introduction because this particular stage is a little tricky. Now first of all, it is formed in the inner membrane or cristae of mitochondria. So if you look at this diagram here, this is the outer membrane, this is the inner membrane. This space in between is called the intermembrane space, and this is the mitochondrial matrix inside. So you will see pictures like this all the time, but usually they don't draw the outer membrane, they just draw the inner mitochondrial membrane here. Anyways, in the inner mitochondrial membrane, there is a high release of electrical potential energy. Why? Because there will be a lot of electrons transported there. And it is for the production of ATP, as you will see later on. Anyways, as we said, there are a lot of electrons are being transported here, and actually these electrons are transported by electron carrier molecules. You can see here there's 1, 2, 3, 4, 5 different electron carrier molecules, and together they make up the electron transport chain, or in short we call it ETC, electron transport chain. Other than the electron transport chain, there is also ATP synthase, which is a membrane protein. It is an enzyme as well as a transport protein, you will see later on. And it basically synthesizes ATP as seen in its name, right? ATP synthase. If you look at it up close, it will look like this. It looks like a little motor. It will spin, and later on I have an animation that will show you how exactly it spins. Okay, so here are the steps for oxidative phosphorylation. Step 1, hydrogens are removed from reduced NAD and FAD. Now if you remember, we said just now there is a lot of reduced NAD and FAD formed in the previous 3 stages: glycolysis, the link reaction, and the Krebs cycle. And this happens at the inner mitochondrial membrane, and this reaction would be considered a dehydrogenation or oxidation reaction. And of course, it's also catalyzed by enzymes. Every reaction here is catalyzed by enzymes, and in particular, dehydrogenase enzymes. As a result, NAD and FAD is regenerated, and if we were to write them in equations, it looks like this. Reduced NAD becomes NAD plus hydrogen atom. Reduced FAD equals to FAD plus 2 hydrogen atoms. And these FAD and NAD that are now no longer reduced, okay, they can be reduced again in glycolysis and link reaction and Krebs cycle. So it goes back to the matrix, it goes back to the cytoplasm in order to help in those reactions. So now, what happens to the hydrogen atoms? The hydrogen atoms that are removed from NAD and FAD, they are split into electrons and protons, or H+ ions. And different things happen to the electron and the proton. Okay, let's talk about the electron first. The first thing that electron do is that it passes along the electron transport chain. The electron transport chain, as you can see here, is in the inner mitochondrial membrane. And There are actually 4 types of membrane proteins labeled here, 1, 2, 3, and 4. The 5th and 6th element here, they are not proteins and you don't need to care about them that much. But anyway, all these proteins together, they form a functional unit called a respiratory complex, or actually, we don't use that term very often, we just say it's the electron transport chain. But anyways, as the electrons move from one energy level to another, the energetic electrons release energy as they pass through the electron transport chain. So because electrons, they are their energy levels are very high and they're vibrating non-stop, that can supply energy to the membrane proteins here. But what do the membrane proteins do? This is what they do. The energy release is used to pump protons. Remember the protons that has came from the NADH and the FADH in the previous few slides? Yeah, that hydrogen ion or proton is pumped into the intermembrane space across the inner mitochondrial membrane. Okay, so the area down below here is the mitochondrial matrix. The area above here is the intermembrane space. Yeah, so it gets pumped across the membrane, and this actually forms an what we call a proton gradient or electrochemical gradient. Now this is particularly formed because the inner membrane is impermeable to protons, because the phospholipid layer has a hydrophobic core, and ions cannot pass through those hydrophobic cores. And also this is active transport. Electrons are releasing energy in order to pump hydrogens across the intermembrane, across the inner membrane, and therefore this is against the concentration gradient. So yeah, due to these factors, the electrochemical gradient is formed. Now, why do we call it an electrochemical gradient? Maybe you're thinking. Now, it's because proton is not just a chemical, it's not just a molecule, it also has charge. So we can expect the intermembrane space to have a much more positive charge because of the high proton concentration than the matrix, which has a low hydrogen ion concentration. Okay, so again, NADH and FADH2, they sort of get dehydrogenated. Hydrogen atoms remove. Hydrogen atom becomes H+ and electrons. Electrons pass through the electron transport chain, and the hydrogen ions use the energy release to be pumped across the inner membrane into the intermembrane space. Now realize in this diagram that NADH seems to be dehydrogenated at complex 1, whereas FADH2 is dehydrogenated at this number 2 membrane protein here. And although this is very often seen in diagrams, it doesn't matter as much. So let's move on. Okay, so where did we stop at? We stopped at the formation of a proton gradient or electrochemical gradient. So what happens next? What happens next is protons move down the electrochemical gradient. So there's a proton gradient, and therefore it moves down via facilitated diffusion. Naturally, no need energy here. But what it does is that it goes back to mitochondrial matrix, and it helps ATP synthase, provide energy for ATP synthesis. This entire process of using an electrochemical gradient in order to make ATP is called chemiosmosis. How cool is that? Anyways, as this is happening, what is the ATP synthase doing actually? So when the protons move down, the electrochemical gradient, the ATP synthase actually rotates. And when it rotates, ADP and inorganic phosphate forms ATP. And ATP, of course, is synthesized. To be precise, the movement of 3 hydrogen ions back into the matrix forms 1 ATP molecule. Let's talk a little bit more about ATP synthase before we move on here. Now, ATP synthase is a very well conserved enzyme, which means that in animals and in us and even in flies or even insects, the enzyme found in the mitochondria ATP synthase is very much the same, and it's very essential to our living, isn't it? Because it produces ATP. And it's fascinating because, number one, it looks amazing on GIFs, and also that it has multi-functions. Okay, it is a transmembrane, so transmembrane means it spans across the phospholipid bilayer. It's a channel protein, and as I mentioned, it's also an enzyme. It has other names like ATP synthetase or stock particles, and you might see some of these names in the mark scheme. Don't panic, it's the same ATP synthase. Just stick to one, okay? Just stick to ATP synthase. Now, it is found in the inner membranes of mitochondria, as you may already know, but it is also found, surprisingly, in the thylakoid membranes of chloroplasts. We will learn more about chloroplasts and photosynthesis next chapter, but spoiler alert, as you know in chapter 1, chloroplasts also produce ATP. Let's go back talking about ATP synthase. ATP synthase structure looks a little bit like this. There's a channel protein on the top, and the enzyme is connected to a part of it in this bottom area. And it's actually able to rotate as hydrogen ions pass through, as we previously mentioned. This is more detailed. It has actually 3 binding sites for ADP and inorganic phosphate, as such you can see here. Okay, there are 3, 1, 2, and 3. And as it rotates, the enzyme binding site changes structurally, changes in conformation to allow 3 different processes to occur. The first thing is the binding of ADP and inorganic phosphate, as you can see here. Number 2 is forming tightly bound ATP, pushing them together. And number 3 is to release the ATP, as you can see here. I think the more you stare at this diagram, the more it makes sense. But don't worry, they don't expect you to draw this spinning diagram or anything like that. You can just stick to the points here. Okay. So enough about ATP synthase. Let's talk about the final step. This is a spoiler alert, but I was gonna say we have hydrogen and we have the electrons. They have to go somewhere, right? They don't just hang out in the electron transport chain forever. Hydrogen ions cannot just accumulate in the matrix. There must be a final hydrogen acceptor and a final electron acceptor. And the molecule who plays a big role in this is oxygen. Oxygen molecule combines with 4 protons and 4 electrons to form 2 molecules of H2O. It forms water. So previous electron carrier in ETC can be reduced again. Without oxygen, all these processes will stop. Okay, you must have a final electron and hydrogen acceptor. With it present, it can ensure electrons keep flowing along the electron transport chain. The role of oxygen and the essential role of oxygen in this process is why it's called aerobic respiration, is why we need oxygen in our cells in order to produce energy. This is its role. It doesn't come into play in stage 1, stage 2, and stage 3, but it comes into play in oxidative phosphorylation. Oxygen is also why it's called oxidative phosphorylation. Maybe there's two sides to this. One is oxygen because it's so oxidative, and also the fact that NAD and FAD, NAD and FAD are oxidized during this process in the first step as well. So just so you can get a better idea, I have this in animation. So we're going to recap a process again here. So we have the electron transport chains and ATP synthase in the inner mitochondrial membrane. Step 1, hydrogen carriers release high energy electrons. Okay, so this is an oxidation process. You have electrons that go into the electron transport chain, and this provides energy for hydrogens to be pumped across the inner mitochondrial membrane. Therefore, there is an electrochemical gradient that's formed. The hydrogen ion will pass through the ATP synthase, and when it passes through the ATP synthase, the enzyme spins and produces ATP from ADP and inorganic phosphate. And of course, the last step, oxygen comes in and removes electrons and H+ ions, becoming water. And that is oxidative phosphorylation. So, summary so far, it happens in the mitochondrial inner membrane again, requires the electron transport chain and ATP synthase, is synthesized from electrical potential energy. ATP is synthesized. Just a list of reactants and products again. Initial reactants is 10-NDH and 2-FADH2. These are products of stage 1, 2, and 3 per molecule of glucose that we calculate just now, and also oxygen as well as ADP and inorganic phosphate. Theoretically, this is the figures, right? 1 NADH will produce 3 ATP, 1 FADH2 will produce 2 ATP. But because some energy may be needed here and there, so on average we calculate it like this. So this is some values you need to remember. 1 NADH will produce 2.5 ATP, 1 FADH2 provide 1.5 ATP. So if you do some mathematics, you have 10 NADH, so 10 times 2.5 ATP plus 2 FADH2 times 1.5 ATP, and altogether you have 28 ATP. And of course, since oxygen is the final electron acceptor and the hydrogen acceptor, it forms water. This is a summary of stage 1 to 4 of aerobic respiration. The following two tables requires you to fill up. So pause this video now, and fill it up. Try to recall what you can and play this video again when you're done. Okay, I hope you filled the tables up. Now let's look at the answers. So in glycolysis, 4 ATP were made but 2 were used. There is net gain of 2 ATP in total. Since this is a balance sheet for one molecule of glucose, the Krebs cycle has turned two times, and therefore 2 ATP is made. There is a net gain of 2 ATP. In oxidative phosphorylation, as we just calculated, there is 28 ATP made for 10 NADH, and 2 FADH2. So add that up together and you get 32 molecules of ATP per glucose. Now, the next table is a summary of reactants and products of each stage per molecule of glucose, and here are the answers. In the exam, I guess the most important parts are not the NAD that gets reduced, but the more important parts are really the glucose and how it forms pyruvate, and how many ATP are formed each stage. So I hope you got that right. So in summary, we can see this picture. There is really two ways that ATP can synthesize in respiration. Number one is substrate level phosphorylation, which we have seen in glycolysis, which resulted in 2 ATP. The link reaction does not produce or require any ATP. The Krebs cycle we produce 2 ATP via the substrate level phosphorylation, but most of the ATP are already synthesized through oxidative phosphorylation. This produces 28 ATP per molecule of glucose. This diagram here also reminds us that glycolysis is happening in the cytoplasm and produces NAD. Krebs cycle and link reaction occurs in the matrix of the mitochondria, whereas the electron transport chain is present at the inner membrane of the mitochondria. Krebs cycle also produces NAD and FAD. And this reduced NAD and FAD produced by glycolysis and Krebs cycle in respective multiple steps is brought to the electron transport chain, where it is used to form ATP. Well, the hydrogen atoms they are carrying helps to form ATP. And that's it. You are pretty much done with aerobic respiration. I hope that was helpful. If you have any trouble and any questions, don't hesitate to ask me via Teams. See you guys soon. Bye!